We interrupt this broadcast to bring you a special report from Cardiff Bioscience News.
We're just going to take you into one of the Cardiff University labs where we've got amazing footage of cells moving. We're going to be talking to one of the scientists, Chris Towers, who's found this out. Here he is, Mr. Chris Towers.
Wow, fantastic. Come over and see. Hello and welcome to CBS News. I'm Mark O'Brien and I'll be our anchorman tonight. We're very lucky tonight to be able to talk to some of the researchers in the field of cell movement. But first of all, we're over to Dami for an introduction to cell movement.
Thank you, Mark. Cell movement is a highly dynamic activity essential to a variety of biological processes, such as the development of an organism, wound healing, and immune response. Wound healing requires neutrophils and macrophages to move to the wound site to kill microorganisms that cause infection. Fibroblasts also move to the site to remodel damaged structures. These examples of cells usually reach their target location via cell crawling. Other examples of cell movement include the swimming motion of sperm cells towards eggs and the power stroke contraction of muscle cells. Cell crawling, however, appears to be the most common mechanisms by which most eukaryotic cells move. All of these mechanisms will be covered in today's new bulletin.
Thank you, Dami, for that most riveting report. But now over to Chris Towers on the lowdown for cell crawling.
Moving cells are orientated by extracellular signaling molecules which cause the activation of the ARP23 complex, which in turn causes profilin to assemble an actin ATP at the leading edge. This pushes the membrane forward. Currently, there are three main methods of explaining how polymerization of actin pushes the membrane forward. They are the elastic Brownian ratchet model, the tethered elastic Brownian model, and the autocatalytic model. In the elastic Brownian model, when the actin filaments, which grow at 70 degrees, make contact with the membrane, the actin filaments bend, which provides an elastic force. The tethered model is very similar, but involves the attachment of actin directly to the membrane. The autocatalytic model is completely different and has to use varying concentrations of actin ATP to explain why the membrane moves forward. The newly formed filopodia then attaches to the substratum at a specific point. It is not currently known how exactly the cell adheres to the substratum and is a hot research topic. The adhesion helps the cell anchor onto the substratum for further movement. And once the cell has moved past this point, it is not known how the focal adhesions dissociate. The last part of the process of cell crawling is the moving of the cell body in the direction of the lamellipodia. This is done by the myosin contracting the actin filaments at the front end of the cell. This moves the cell body forward so that the cell goes in that direction. This similar thing happens at the rear end, where the myosin contracting moves the rear end forward towards the front end.
Thank you, Chris. And now for some sperm. Over to the specialist, Emily Kelly.
Sperm is a good example of a single eukaryotic cell movement by the mechanisms of flagella. There is a head, middle piece and tail part to the cell. However, it is the middle piece and tail that supplies the cell with the force of movement. The middle piece is packed full of mitochondria, which supplies ATP needed for the sperm's journey through the cervix, and the tail region, also known as flagella, provides the beating motion in order for it to swim. The flagella is a projection-like extension of the membrane which is capable of being flexible, as its function is to propel the cell forwards. The organisation of the flagella is very specific, and it is seen across all types of eukaryotic cells that contain flagella, possessing a central bundle of microtubules called the axonem, in which 9 outer doublet microtubules surround a central pair of singlet microtubules. The characteristic 9-2 arrangement of microtubules is seen when the axonem is viewed in cross-section. Each doublet microtubule consists of alpha and beta tubules. The alpha tubule is a complete microtubule with 13 protofilaments, while the beta tubule contains just 10 protofilaments. The bundle of microtubules comprising the axoneme are joined to each other by nexin links and is surrounded by the plasma membrane. Regardless of the organism or cell type, the axoneme is about 0.25 micrometers in diameter, but it varies greatly in length, from only a few microns to more than 2 millimeters. It is the interaction of the alpha and beta tubule with dynein arms that represent the force behind the sperm's motility and is a true representative of the power stroke. It is a four-step process. 1. The dynein arms anchored to the alpha tubule attach themselves to the binding sites on the beta tubule, which is located above it. 2. Power stroke. The dynein arms undergo a conformational change that causes a forward movement of the lower alpha tubule, which slides towards the minus end of the beta tubule. 3. The arms now have to detach from the beta tubule in order for another power stroke to occur. 4. Before another cycle can happen, the dynein arms reattach to the upper beta doublet tubule.
Thank you, Emily. You're most knowledgeable on that subject. For now, we're going over to Craig, who's going to talk to us about muscle contraction.
One type of cell highly specialist for movement is a skeletal muscle cell. A skeletal muscle cell, or muscle fibre, is a multinucleic cell made up of myofibrils. Each myofibril is made up of microfilaments. Actin is the thin filament and myosin the thick filament. It is these microfilaments which form sarcomeres, the contractile units of a muscle cell, and give it its striated appearance. The Z-lines mark the boundary between the adjacent sarcomeres. They are made up of proteins called actinins. Attached to these are the actin fin filaments which extend towards the M-lines in the centre of the sarcomere. The thick filaments protrude from the M-line towards the I-band between the fin filaments and are attached to the I-band by proteins called titin. Titin keeps the filaments in alignment and stops the muscle fibre from overstretching. The area where the thick filaments are between the fin filaments is called the zone of overlap. In this area, each fin filament is surrounded by 3 thick filaments, and thick filament by 6 fin filaments. It is in this zone where the filaments slide past each other, causing the sarcomere to contract. This is called the sliding filament theory of muscle contraction. Now to take a closer look at the forces which cause this movement to happen. Muscle contraction produces tension, which is a form of energy, thus requires energy expenditure to produce this. This energy is provided to the sarcomeres in the form of ATP. This picture is a representation of the actin and myosin II filaments in the zone of overlap. The myosin II filaments have heads, and the actin has troponin running alongside it, covering its active binding sites, which the myosin heads will attach to. When calcium enters the sarcoplasm after a nerve impulse, the calcium binds to the troponin and uncovers the active site. The myosin head is then able to reach it and bind. The myosin head is cocked backwards like a spring by the energy released from ATP when cleaved into ADP and an inorganic phosphate. This energy also helps the myosin head to bind with actin's active site and form a cross bridge. When the myosin releases the ADP and phosphate, the head, which is still bound to the actin filament, is released and pivots towards the M-line, pulling the actin filament along with it. This movement is called a power stroke. When another ATP molecule binds to the myosin head, the actin is released, which re-exposes the active site. This ATP is then hydrolyzed, and then the energy from this recocks the myosin head, ready for another cycle. This process is repeated several times a second in the sarcomere, whilst calcium levels remain high enough to expose actin's active site and there is sufficient energy in the form of ATP. It happens in all the sarcomeres which make up a muscle at the same time, and each power stroke causes a sarcomere to shrink by 0.5%. As multiple myosin heads are bound to the actin at any one time in each sarcomere during contraction, when the myosin head releases the actin, it is bound to the other cross bridges still, so it still holds the actin in place, so that when another power stroke happens, it contracts the sarcomere more and more until the muscle stops contracting or the titin prevents overcontraction.
Thank you, Craigie boy. And now over to Matty Matmad for a summary.
Thank you, Mark. So, why do cells need to move? Well, there's chemotaxis, which is the movement of cells towards biologically useful molecules, high concentrations such as food molecules or glucose. Biological role in multicellular organisms. Certain cells, such as platelets, as well as muscle contraction, have specific activity within tissues and throughout the body to carry out their intended effects. So, how do cells move? Well, cells can move in many different ways. One of the main methods that they can move in is crawling, and there are three main stages. Initially, it's a propulsion of filopodia by orientation and extension of the actin network by the involvement of the protein profilin. Secondly, the filopodia must adhere to the leading edge of the cell. And thirdly, the contraction of the actin microfilaments from the negative end of the cell pulls it in a specific direction. So the propulsion of filopodia through the orientation and extension of the actin network by the protein profilin. Secondly, the filopodia must adhere to the leading edge of the cell. And lastly, is the contraction of the actin microfilament from the negative end, which causes the pulling of the cell in a certain direction. So what are the main forces involved? Well, the main force involved is the extension of the actin microtubules from the positive end pushing the cell membrane out, as well as tension across the cytoskeleton from the leading edge all the way through to the tail, anchoring the cell. Cell swimming. Dying undergoes a conformational change, which pulls the A tubule towards the basal end of the B tubule. Now for a specific example of cell movement, a skeletal muscle cell. The forces involved with muscle contraction of skeletal muscle cells are due to the tension locked up in the myosin head, as well as the torque produced by the swivel of the same head group upon the hydrolysis of ATP, named the power stroke.
Back to you, Mark. And that is all we have time for today. I've been Mark O'Brien, and I love science. Stay classy, Cardiff.
I like to move it, move it. I like to move it, move it. I like to move it, move it. You like to move it, move it. All girls all over the world. Original King Julian for the case man. I love all the